Next, I'd like to introduce John Trojanowski, who is the William Malmese Truman G. Schnabel Jr., M.D., Professor of Geriatric Medicine and Gerontology, Director of the Institute on Aging, Director of the Alzheimer's Disease Center, Director of our new Udall Center that we're all very proud of here, as well as co-director of the Center for Neurodegenerative Disease Research. John is going to be talking about new drugs and new treatments for FTLD and related conditions. Thanks, John.
Thank you. Thank you very much, Murray. And Beth, that was fantastic. The previous speakers have made my job a lot easier, and I think you can appreciate just from the type of people that have spoken this morning, Beth included, that we're wonderfully privileged to have a great group of collaborators in the work that we're trying to do on frontotemporal dementia. I was listening to the talks and had a few thoughts that are not part of my formal lecture, but I just remember when Virginia Lee and I started our work here at Penn 20 or so years ago, and I'd go to support groups and on Alzheimer's disease, and people would ask me about therapy and when we'd see drugs and that would benefit people. And as a physician, this was very depressing because I couldn't say very much about what to expect in the future. And that was just 20 years ago, and now there are over 60 clinical trials, 60 different drugs in clinical trials for Alzheimer's disease. So even though, yes, you can't hear me? Okay, I'm sorry. So even though we're still on a steep learning curve for frontotemporal dementia, I'm more optimistic than ever, some of the progress you've heard about already this morning, that justifies that we're on our way towards identifying targets. Targets are the things that you aim drugs at to slow disease, to arrest disease progression or prevent it. And in fact, there's even optimism about prevention. The Alzheimer's Association and the CDC just collaborated over the space of about 2 years to put together a number of recommendations. I'm proud to say that Kathy Jadrewski, who's in the audience here, represented Penn in this effort, recommendations about successful aging, successful brain aging, so things that you can do in your life to make a difference in how you age. And they're things that you're all familiar with. Maybe your grandmother told you about them. Read a book, keep your mind active, be physically active, eat your spinach, your leafy green vegetables, blueberries, and so forth and so on. So there's a list of things that we can recommend, almost certainly urge you to consider in your daily life that may help you age successfully. Many of the things that are good for your heart are also good for your brain. So the other thing to point out is that you've heard a lot about diseases, and it sounds pretty scary getting older, obviously, with all of these aging-related diseases. But there is a lot of good news. There's been studies over the last 20 years showing that disability rates are going down for people over 65. So it's not all medicine. It's part of what we're all doing in our lives to live better. Wearing our seatbelts, not smoking, exercise, and so forth. So it's although we've heard about some difficult conditions, it's not all dark clouds and bad news. I'm here to tell you that there is a lot of reason to be optimistic, even for a very challenging disease or group of diseases like frontotemporal dementia. So this is a slide that is intended to summarize. Let me see if I have a pointer here. Oops, didn't mean to turn it off. It's back on again. Here's a pointer. Okay, sorry. Yep. Good. So these are the most common aging-related neurodegenerative diseases of our time. And what I'm showing are the common names of the disorders over here on the left. These are the abbreviations for the pathologies that help us diagnose them. You've heard about Lewy bodies, LBs, neurofibrillary tangles, senile plaques, and so forth. And the great revolution is that we're able to name the proteins that make these inclusions, and that gives us traction towards drug discovery. And the latest kid on the block you've heard about already is TDP-43 in frontotemporal dementia. It may explain about half of frontotemporal dementia. And people sometimes look quizzical when I tell them that these diseases are protein misfolding diseases. They say, what the heck is that? So my best way of trying to explain that, here's today's agenda. It's on a piece of paper. And this piece of paper conveys information. Proteins convey information. They're signaling molecules. They tell the cell to take up glucose or whatever to function normally. And when I go like that, I've just misfolded this piece of paper so it's lost its function. It doesn't work anymore. I can't read it because it's folded. Now, the good news is, and I'll show you this on a schematic, is in all of our bodies we have mechanisms to unfold misfolded proteins. And when that works and everything is hunky-dory and we're home free. And if they don't unfold properly, then you can take this and put it in a trash can and then it's out of the way and it gets cleared. If that doesn't happen, then we've got problems. And that's what I want to show you in the next slide here. We use the analogy, my wife and I, Virginia Lee is my colleague, we bike to work every day, and when I bike over the South Street Bridge, if my bike stops, whatever, for whatever reason, not deliberately, and it's broken, I've got to get off the bike and then check it out. Has the chain come off? Or do I have a flat tire? The diagnostic is very important. Finding out why my bike is broken is the first step in fixing it so it can get back on it again and sail on. And so the point I want to make is that we found out an awful lot about how the brain is broken in Alzheimer's disease, and we're making the same progress now in the group of disorders called frontotemporal dementias. So this is the schematic that I referred to that displays a lot of mechanistic understanding. It's very complicated, very technical. I'm showing you slides that I show my colleagues, by the way, so I'm hoping I'll be able to explain them in non-technical language, but this is the same sort of material that I use when I go to conferences and talk to my peers. So up here are the proteins that I referred to that are normal, functional. Tau is it's a good guy. Tau is a good guy, it's not a bad guy. When it gets misfolded, then it's a bad guy. TDP-43 we know less about, but we think also it has important normal functions. When these proteins misfold, they form these chunky deposits, and that's what accumulates and floods nerve cells so they don't function normally. Now, fortunately, we have the garbage disposal system here that I referred to, the proteasome, the lysosomes that under normal circumstances would sweep all this debris away and we would be okay. Or we have the chaperones that, just like a chaperone that you're familiar with, keeps an eye on your kids at the school prom or whatever, make sure they don't get into trouble. These chaperones do the same thing, making sure that proteins don't misbehave and get into trouble. The bad news happens when this process goes in this direction. You can't reverse the misfolding. You can't get rid of the misfolded products, and they accumulate as, in this case, neurofibrillary tangles in an Alzheimer brain or in a frontotemporal dementia brain. And then a number of downstream consequences of this are depicted here, and a lot of this is, again, very technical. But oxidative stress, that's rust. You can think of that as getting your proteins rusty so they don't function properly. These are the energy machines in cells, the mitochondria. If they don't work right, you're not going to have energy to have your nerve cells working properly. I'll mention a little bit about axonal transport. The transportation systems and highways in the brain are very important to keep cells alive and for communication. You can have changes in how genes are activated. You have a form of cell death called apoptosis. So there's lots of things that we know that are bad that happen when this process goes in this direction. And that gives us a roadmap to think about how to stop these processes for therapeutic benefit. And so here's just we don't know a lot about TDP-43. You've heard about it already mentioned a couple times. It's just the name of the protein, the TAR DNA binding protein with a molecular weight of 43. That's where it comes from. That's where it comes from. It's a mouthful, obviously. Maybe we'll come up with a better name for it in the future. But normally it's in the nucleus where it does important stuff, all of which we don't know about, but in regulating how genes are expressed, turned on, turned off, to make different products that cells need. The bad stuff happens when this goes out of the nucleus and deposits in the processes of cells or in the cell bodies, or if it clumps inside the nucleus itself. And we don't know all of the deleterious effects of that, but we will focus our therapeutic efforts on how to get the genie back in the bottle, how to get that protein back in the nucleus so it performs its normal functions instead of depositing inappropriately in the wrong place. That's about all I can tell you right now about TDP-43, but knowing the miscreant protein is the beginning, is the first step towards drug discovery. And I'll give you some examples of that when we talk about tau. I also want to emphasize how important it is to work on Alzheimer's disease, ALS, to look at all of these side by side. We learn an awful lot about them, and we've heard about some of these other diseases. There are lessons that we can learn. If we could solve mad cow disease, maybe we'd be able, which is very, very rare, of course, maybe we'd be able to solve frontotemporal dementia and Alzheimer's disease. The other reason that we need to know about these other diseases is they can masquerade, and you've heard that already. You've heard how Alzheimer's disease, actual Alzheimer's disease, can look like frontotemporal dementia. Well, in a clinical trial, or when we have drugs that are disease modifying, we want to be able to sort out these patients and treat them with the right drug that's appropriate for Alzheimer's disease. And I use the analogy of pneumonia. Pneumonia looks the same whether it's caused by TB, pneumococcal pneumonia, or any of 100 other bacteria, but the name of the game is finding out the bug so you can give the right drug, and that's the way we think about these neurodegenerative diseases as well. So it is important to study them side by side for those reasons. And as I mentioned, when you take away Alzheimer's disease, about half of frontotemporal dementia is caused by tau mismetabolism and the other by TDP-43. So this is a very technical slide, and just bear with me for a moment. But it outlines 7 different targets for drug discovery. I mean, for me, this is breathtaking, because because I couldn't even begin to tell you what the problem was 20 years ago when I went to support groups and said there's something bad going on in the brain, but we didn't know it was tau until 1991. That was a big discovery that came from the University of Pennsylvania. So it is breathtaking to be able to get up in front of you and say, I've got 7 ways to cure tau pathology. I mean, that is just very exciting for a scientist, and I think it must be for patients and caregivers. So here's a nerve cell. Tau plays a big role in keeping these microtubules polymerized. And if you don't remember anything else, this is the transportation highway of the nervous system. Very important to keep this intact so that traffic moves up and down appropriately, keeping the nerve cell alive. So a couple options to consider. We can get rid of the bad tau. We can figure out a way to suppress tau. We can figure out a way down here to inhibit tau phosphorylation. What does that mean? Well, that's just a modification of the protein that regulates the on and off binding of tau to microtubules, and tau seems to get too phosphorylated in Alzheimer's disease. You can then try to maintain the normal conformation of tau. Remember, this is keeping it folded properly. So maybe we could figure out to replace the defective chaperones that aren't doing their job so that tau folds properly. We can stabilize microtubules. We can make the trash compactor work better to get rid of the debris in the brain that's formed by tau. Or we can do something called anti-aggregation. We can give compounds that help the chaperones refold tau into the proper position. And finally, there's neuroprotection. I'm going to tell you a little bit about that from some recent exciting experiments from our group. If we don't do this for tau, these are the consequences. So here are the train tracks, here are the cross ties. This is tau, keeping the train tracks in shape so that the train hums along properly. And if that doesn't work out in the brain of in a nerve cell inside of an Alzheimer patient or a frontotemporal dementia patient, then you get train wrecks and you get toxic waste dumps. So that is the problem in all of these neurodegenerative diseases, including tauopathies and TDP-43 associated frontotemporal dementia. You have toxic dumps of protein that accumulate, clog up nerve cells, and lead to their demise. So our job is to try to figure out how to reverse this process. And fortunately, we're learning an awful lot about the process so we can begin to conceive of therapies. So back in 1994, if you can read the data up here, in 1994, we thought we knew enough about how to fix tau mismetabolism that we proposed we might be able to use microtubule stabilizing drugs. Now, even though we had the idea, we didn't have animal models. We had to figure out a way to test our idea. And it wasn't until 1999 that we had an animal model to test this idea in. And there are lots of drugs that stabilize microtubules that are used already in medicine. You may know paclitaxel, which is used, or Taxol, which is used for cancer. Now, wouldn't it be phenomenal if we could take an FDA-approved drug and bring it online for patients with Alzheimer's disease? Well, we published a study showing proof of concept in an animal model in January 2005, and this is some data from that study, and don't be intimidated, I'll be able to walk you through it very easily. We were able to show that by giving Taxol, we could reverse the loss of microtubules and the fast axonal transport problem, that the impediment in transport that took place in these mice. And you don't have to be a biochemist, I think, so just focus on this. This is real biochemical data, so you'll be able to walk home tonight and tell your kids, your family, I learned some biochemistry at that meeting, and I know some biochemistry about Alzheimer's disease. So what we're showing here, SC is spinal cord, and these are millimeter increments away from the spinal cord. So spinal cord is here, going down my arm or down my leg would be incremental centimeters, of course, but in a mouse it's a few millimeters. And you can see that we've labeled the proteins in the spinal cord of this mouse, and you can see that the sham-treated mouse, the proteins haven't moved that far to the right. That's because they have an axonal transport defect because tau is not working properly. That's what goes on in people, we believe. And when we give our drug, you can see the proteins move much further to the right. We've fixed the transport problem in these mice, and in fact, we showed their behavior improved. So that's called a proof of concept study. And right now, and this just summarizes what I've said, we were able to improve the pathology and the behavioral impairments in these mice. And right now, we are intensively working on this list of, not all of them, but many of the natural products and derivatives of natural products that have the same function as paclitaxel. They come from a wild assortment of critters out in nature. And the good news is the pharmaceutical industry is very interested in many of these natural products because they're used to treat cancer. And so we hopefully will be able to build on that pharmacology to understand how to get these products into the brain better. So that's the problem that we're facing right now, engineering these molecules to enter the brain so that they would be effective in the brain, which is where they have to be. For cancer, they don't have to go into the brain. But this work is in progress. I'm pleased to say that we've done other studies, so we're able to cure now the pathology of Alzheimer's disease and frontotemporal dementia in mice. This was not even a dream that I thought would come to pass in my lifetime when I started working on these diseases 20 years ago. So this is a phenomenal progress, and it's not just our laboratory. Other laboratories also are showing proof of concept studies in animals that, again, is the beginning of a conversation with the Federal Drug Administration, the FDA, about how to move a drug out of the laboratory and into people. And so the closing minutes, I'm going to talk to you about another similar success story, again in mice, with a drug called FK506. We just reported this in the journal Neuron. And I'm going to show you some of the actual data again, so you'll be able to go home and tell your family that you've learned some neuropathology, and you've actually can explain to them what a proof of concept study is. So here's the mouse. It has the pathology of frontotemporal dementia caused by tau. You can see the tangles. This is the mouse brain here. It's a big, the mouse has a very little brain, of course, but we've blown it up in this slide. And all of these dots are the tangles that accumulate that we've been able to engineer these mice to develop because we put in one of those tau mutations that you just heard Beth talk about. And when we do that, we can make the mice develop really bad disease. There's a lot of atrophy, a lot of neuron loss that's occurred in this mouse brain. This is a very complicated slide. I'm not going to walk you through each of the panels. I just want to point out that this is the untreated mouse brain up above here, and you can see there isn't a lot of blue, meaning it's lost lots of nerve cells. It's the untreated mouse, and it's also shrunk an awful lot. And if you look over here, you can see the tau pathology. Now down below is our brother-sister littermates of these exact same mice, but we've treated them with a drug called FK506. You may have heard of it. It's used for to block organ rejection in people. So it's given all the time to people who get a liver transplant, a heart transplant, a lung transplant. So we know a lot about the pharmacology of this drug. This is a big step, a big advantage in trying to bring a drug into a clinical trial for patients with other diseases. And what I want you to focus down here is the survival curves for these mice. So the mice that were not treated, that's the red line. So these are the mice that weren't treated by FK506, and about 80% die by 1 year. The FK506 treated mice, and we just tried one dose. We've got to go back and try different doses now. But just with one dose, we had the good fortune to pick a dose that wasn't toxic, that had beneficial effects, it prolonged survival, and eliminated any of the pathology that we saw in the transgenic mice, the engineered mice that had the tau disease like frontotemporal dementia. And this is a summary of what I've just said. I don't think I have to read through it all. This is a lot of technical speculation about how the drug works. We don't know if it works through blocking inflammation. I didn't point that out on the slide, but we were able to suppress the inflammatory consequences of neurodegeneration. That is a part of almost all of these neurodegenerative diseases. We're able to suppress the tau pathology, whether that's due to a different pharmacological action or is linked somehow to the inflammation. We don't know for sure, but we are now, again, at a point where we can have a conversation with the FDA about how to bring these types of compounds to clinical practice. Of course, I'm talking to Murray Grossman about this. We've just had a couple conversations with folks at Penn here about how to create an IND, an investigational new drug application that we could submit to the FDA so that with a few more animal studies, we'd perhaps be in a position in 2 years or so to bring to launch a clinical trial. I want to emphasize how important your advocacy is. Your getting behind the Frontotemporal Dementia Association is very, very important, and partnering with other organizations to make it known to the world how important these diseases are. And a lot of us in science are doing the same thing too. Here's an article by Jean Marks. It just appeared last week. It's actually going to be one of the handouts. It's out on the AFTD counter. And what Jean Marks is saying briefly in her article is, okay, we're going after A beta in Alzheimer's disease, but what about tau? We've neglected tau. So it's important to realize that we don't know which of the targets is going to be most meaningful to hit with the drug. We can't predict that. Although I said we're curing animals in the laboratory of these pathologies, we all know that this is not like a computer-assisted architecture where you can do a schematic of how you build the Bank of China or the Empire State Building or whatever, and you know for sure that the building will go up and it'll be done just exactly as you design it. Clinical trials are messy. Clinical trials are not as precise as other types of science. People actually have toxic reactions and on some occasions die in clinical trials. So these are very, very serious human experiments that have to be done under, obviously, ethical conditions and so forth. But we would never be in a position to talk about bringing a new drug into clinical trial in people unless we had success in animals. And I'm telling you that that is what is happening, and that's why Jean Marks wrote this article in Science to let more people know about the potential of tau as a target for drug discovery for Alzheimer's disease, and it's also true in frontotemporal dementia. I'm pleased to say that working with the AFTD group and a number of other scientists, some of whom are here, including Murray, and the National Institutes on Aging and the National Institutes of Neurological Diseases and Stroke, we had a meeting last January where we brainstormed about future directions for a number of things related to frontotemporal dementia, including drug discovery. And I'm pleased to say that this consensus report of the group has just been accepted in a journal. It will appear in printed form sometime around January. I believe this is also a copy. You have an abbreviated copy of this on the AFTD bench outside or desk outside. If anyone wants a copy, I'm pleased to give it to anyone who wants it. Again, it's a technical document and goes into scientific detail along the lines of what I'm showing you here. So these terms will not be familiar to you, and I don't expect you to know them, but I'm just pointing out a list. This is the list of candidate drugs that we can think about for frontotemporal dementia. I couldn't even imagine a list like this 10 years ago. So this is the progress that is emerging from laboratories. I've talked to you about FK506. I've talked to you about a number of things related to tau pathology that we are doing today in the laboratories that we're hoping to bring to clinical trial in people. And this is a more detailed summary of the agenda that we think should be supported by increased funding from the federal government to the NINDS, National Institute of Neurological Diseases and Stroke, NINMH, NIA, National Institutes of Aging and Mental Health. And again, I don't expect you to read through this and understand that all these are technical technical recommendations for how to move the field forward so that we have better diagnostics and better therapeutics for frontotemporal dementia. And I believe that is this is my last slide now. I want to show you what we're doing at Penn. You've met a lot of my colleagues already. You know that we work together very synergistically as a team. We have established a drug discovery program in our Center for Neurodegenerative Disease Research. This is a $1.5 million robot. These tools are not cheap, but we have a robot now that enables us to screen thousands of drugs a day on targets that are relevant to the pathways that I've showed you that lead to disease in people. So the first step is to screen with the robot in a test tube-like situation, except what we're talking about is 1,536 wells, so a miniaturized form of a test tube that enables us to screen thousands of compounds in parallel simultaneously. And then we take the hits, the compounds that seem to do something in a test tube or in this robotic format. We then can modify them with our collaborators from medicinal chemistry, Amos Smith, Carlo Ballatore. They can figure out ways to make the drugs enter the brain better, be less toxic, and so forth and so on. And then we go to animal studies, and we get to the position that we're in with FK506, which I've shown you, or paclitaxel, and then we can start talking again to the FDA about how to move into people. So I will close now and tell you that this is the most exciting time to be doing science on dementias that affect millions and millions of people, and I'm hoping in my lifetime we'll see drugs that make a difference. Thank you very much.